Hello, I'm Larry Risen, President and CEO of BioTrak Market Intelligence, a leading provider of REMS assessment design and implementation consulting services. We're excited to bring to you a three-part series on strategies for REMS assessments. So welcome to part 2. In this video, I will discuss assessment protocol design, review sampling considerations, address recruitment of stakeholders, and explore sample size and statistical considerations. So let's discuss some specific techniques for developing and implementing a REMS assessment protocol. This will include description of stakeholder sample recruitment methods, rationale for the sample size, description of how sample bias can be minimized, statistical analyses and reporting metrics, performance targets, and the survey instrument itself. The protocol design should describe the methodology for surveying key stakeholders and analyzing results. So first, let's review how to design a survey program that recruits stakeholders in a manner that achieves a representative sampling while minimizing selection bias. This is one of the greatest challenges facing a REMS assessment and is particularly difficult when doing patient recruitment. Consider this: your REMS assessment should be a reliable prediction of actual market behavior based upon prescription events, meaning it should predict performance of the REMS at the level of filled prescriptions. Well, we all know that healthcare providers vary greatly by prescription volume of the drug in question. A healthcare provider sample should weight preference towards higher volume prescribers since they make up a larger portion of the prescription universe, and hence have proportionally greater public health exposure by virtue of the number of drug-treated patients they see. Specific know-how for prescriber sampling techniques requires both experience and expertise in working with a range of third-party data sources. So if I can summarize, the ideal representative sampling will have, for example, decile 10 prescribers and their patients comprising about 10% of the survey sample, decile 9 prescribers and their patients comprising the next 10%, and so on. Following this method, deciles 3 through 10 comprise about 80% of the survey sample. In order to achieve this sampling mix, special techniques must be utilized to ensure sample recruitment methods reflect this decile weighting over a series of repeated measures. Since there are fewer prescribers in the top deciles and a greater number of prescribers in the lower deciles, a larger share of the top decile prescribers will be represented in the survey. For a prescriber stakeholder survey, we suggest restricting your prescriber and patient sample to those who are defined in the REMS as a target for the communication program. The methodology for recruiting key stakeholders should result in achieving a very similar sample with each assessment reporting period. Remember, this is a repeated measures study design. This requires some thoughtful planning during the protocol development. Many pharmaceutical companies are exasperated by the thought of recruiting patients for a REMS survey, and for good reason. Reaching patients in a manner that achieves a representative sample of prescription events for a particular drug is very difficult and an expensive undertaking, but nonetheless necessary. In our experience, engaging representative prescribers is a starting point for recruiting patients. For unique patient populations, it may be necessary to reach out and use other methodologies, such as utilizing support from patient advocacy groups, patient registries, registrations through a website for a particular product that that company has, or other organizations. Print and radio advertisement is also an option, but can be very costly and yield limited results. Now let's discuss sample size. We highly recommend that you power the study according to the performance thresholds for individual measurement domains of interest. The statistical plan The expected measurement range and the desired sensitivity will determine the sample size needed for reliable analyses. Our preferred statistical method for measuring comprehension and understanding of product risk and for measuring communication reach of a REMS element is a binomial test. Pass, fail, yes, no, correct, incorrect. These measurements allow us to take a test score and convert to a binomial statistic or measure incidence rates. We have found that reportable sample size of 150 stakeholders is usually adequate for each REMS assessment reporting period. For example, you might establish a REMS performance goal of achieving patient understanding of risk and appropriate actions at 80%, meaning that at least 80% of the patients scored correctly on some defined percentage of key risk assessment questions. This same approach would be applicable for any stakeholder group that you're measuring and meets FDA's draft guidance for REMS assessments. This concludes part 2 of our REMS assessment video series covering an overview of REMS protocol development and survey design considerations. We will continue in part 3 by looking at specific techniques for implementing a REMS assessment and sharing learnings from past REMS experiences. If you missed part 1, we recommend you go back and review that introductory video. Thank you for viewing this video with us.